Okay, hi everybody. Welcome to the second event of our Making Soils Data Actionable webinar series. I'm Danny Gilardi, Senior Soil Scientist at the Washington State Department of Agriculture, and I am so excited to welcome you all here today to talk about soils data. If you have been working in this space, you've probably noticed that more soil tests and data are available to farmers than ever. And while this can be empowering, it can also be really confusing. New information on soil chemistry, biology, and physics can sometimes be difficult to translate into action. And so the Washington Soil Health Initiative has put together this series to showcase just how useful and necessary soils data can be to making in-field decisions. Every Wednesday at noon this month, we will be showcasing projects from across Washington that are using real data to make real-world on-farm decisions. Today, we have Lindsey du Toit here to help us understand how to make soil biological indicators actionable. Lindsey is a professor and extension plant pathologist at Washington State University and is the current chair of the WSU Department of Plant Pathology. Her vegetable seed pathology program focuses on the causes, epidemiology, and management of diseases affecting vegetable seed production in the Pacific Northwest, primarily in seed crops such as spinach, beet, chard, brassicas, carrot, and onion. And so for everyone in the audience, there will be a Q&A at the end, but feel free to post your questions into the chat feature as we go. You can find the chat feature at the bottom of your screen in a little thought bubble icon. And with all of that, thanks so much for being here today, Lindsey. Feel free to take it away.
Thank you very much. Right. We'll get going on this. Thank you very much. I really appreciate the opportunity to share with you some of the perspective on biological data related to soil health that I'm hoping will guide you in your work. And I'm using a case study of a disease that we have worked on for many years in my program. This is spinach fusarium wilt. And you'll see as we go through this why I think this is a very good case study for looking at biological data. All right, so let's see if I can forward my slides. All right, just to give you a background on why I work with seed crops and why I was hired specifically to focus on diseases affecting vegetable seed production in Washington State. Washington State is a very important region climatically for production of vegetable seed crops of a number of species, and actually not just vegetables, but many species, because of the very distinct climatic requirements to produce high quality seed. So for vegetable seed production, the three areas circled in red here are important for production of seed crops of certain species, such as the crops that Danny mentioned in the introduction. And so these are the two areas that I work primarily, Northwestern Washington and the Columbia Basin. To give you an idea why these are so important, if you look at this map of the world, I've circled in red the regions where spinach seed crops are grown. And these are the only areas of the world that have the right climatic conditions for spinach seed production, because in order for spinach to go to flower and produce seed, you have to have long day lengths, so it needs to be high latitude. You have to have mild summers because it is a heat-sensitive species. It's not tolerant of high summers. And you need dry summers because you don't want high disease pressure from fungal and bacterial pathogens infecting that seed. So these are the only regions of the world that have those three combinations that are necessary for production of spinach seed. And as a result, Western Washington and Western Oregon are the only region in the US where we can grow high quality spinach seed production. So we produce about 20% of the world's supply of spinach seed in Western Washington and Western Oregon. So that seed is moving all over the world from the Pacific Northwest US, and that's really important not to be carrying pathogens with that seed. This is the photo of a spinach seed crop. Spinach is dioecious, meaning it naturally has separate male and female plants, which really facilitates hybrid seed production because you naturally have the separation of the genders. This photo shows the male rows, which are the lighter color, and they, let's see if I can put on my laser pointer. Pointer options. There we go. So the lighter colored male rows versus the female rows. Females don't produce pollen, but there is a small percentage of the female rows that will produce pollen, and those lines have to be rowed where crews walk through and remove any female lines that start producing pollen, because otherwise you get inbred seed. So this is typical. It's a wind-pollinated species, so these crops have to be separated from each other in order to prevent unwanted cross-pollination, which further limits the amount of land that you can use to grow a spinach seed crop every year. But we have the right climatic conditions for this. As I mentioned, these seed crops have to be isolated because of wind, and that creates further restriction on land options. And these crops all get pinned at the beginning of the season to determine where they're located, and making sure that male lines that are close to seed crops that are close together have the same male line. Just so you're aware that other seed crops grown in this area that are really important are, for example, cabbage seed crops, where you can see the photos I'm showing here are the cycle through that 16-month process from producing the seedlings in the greenhouse, planting them out in the field in August, covering them with the winter if it gets too cold, staking them in the spring when they open up and go to full flower, bringing in honeybees, and then harvesting the seed. And that's a 16-month process, and only can be done in Western Washington because these biennial brassica species don't tolerate the cold on the east side of the mountains. But the east side of the mountains is really important for crops like carrot seed production. This is also a biennial. Many of the inbred lines are not cold tolerant, so they grow the roots down in the Imperial Valley of California, dig them up, fertilize them in cold storage, and then transplant them in the Columbia Basin or Central Oregon in the spring. And onion seed crops, which are also a more cold tolerant biennial species that seed is grown from in the Columbia Basin. And these species I work with are mostly small seeded, but the Pacific Northwest is also really important for crops like sweet corn seed, bean seed, and pea seed production. And a really important criteria for the seed industry is to produce seed that is true to type genetically, so making sure these crops are isolated, high quality seed germination and vigor, freedom from pathogens, freedom from noxious weed seeds and other debris, and the ability to compete on the market when the labor price can be pretty high and the cost of producing these crops is very high. And when you look at the demand for seed, this is really interesting. The crop like spinach, there's been just a tremendous increase in the demand for baby leaf spinach because of the popularity of the and convenience and the health of crops like baby leaf spinach. And these baby leaf crops are seeded at populations of 7 to 9 million seed per hectare, with about 3 to 4 million seed per acre. And they're harvested in 20 to 40 days after planting, and they're sequential plantings. So there's a very, very high demand for seed of spinach because of the advent of the baby leaf industry. And you can see the intensity of this production means that a hectare of a hybrid spinach seed crop only produces enough seed to plant 10 hectares of baby leaf. So a 1 to 10 ratio, whether you're talking acres or hectares, because of the huge amount of seed used in these baby leaf crops, which has really increased the demand for seed and put pressure on areas like the maritime region of Western Oregon and Western Washington to provide seed. So even though we have a really ideal climate, one of the big problems is the soils in this area naturally are acid and low in calcium. And these conditions are highly conducive to a soil-borne pathogen called Fusarium oxysprum, Forma specialis spinaceae. So it's specific strains of Fusarium oxysprum that are pathogenic to spinach. So this disease, Fusarium wilt, is very recalcitrant in our acid soils in Washington and Oregon. I took this photo in 2018 of a hybrid spinach seed crop in Skagit County. And you can see the female row is very, very susceptible to Fusarium wilt. She's dying. The male row has some resistance. This field had been out of spinach for 18 years. And growers normally would feel like they try to find fields that have not had spinach for a minimum 10, ideally 15 plus years. But even then, we have scenarios like this where despite 15, 18 years, we see significant wipeouts to Fusarium wilt. And that's been a real challenge to see how can we manage such a recalcitrant disease in this limited area that is suitable climatically for production of spinach seed. So this pathogen is widespread in the soil. It produces these dormant survival structures called chlamydospores. The infection occurs through the fine roots and then gets into the vascular tissue. You can see it blackens the vascular tissue. You can get wilt at a seedling stage or at a mature stage. This lower right pictured photo was taken of a seed crop that had been out of spinach for 15 years. And again, you can see that female row is, the female line is really, really susceptible. And then when a spinach seed crop is harvested, it has to go through full senescence to get that seed to finish maturing and drying. And all those dead residues get colonized by Fusarium. I'm not sure if you can see this faint salmon-colored base to the stem, and that's just full of thousands, hundreds of thousands, millions of spores, shown in this lower left picture, forming on that senescing tissue, because Fusarium is a really good, what we call a necrotroph. It likes to feed on dead tissue. So you get this massive increase in the amount of spore production when these seed crops are drying down, and all that gets incorporated back into the soil and adds a huge load of inoculum. So, there's been a real question about for growers, how do we manage this? Is there a way to reduce that rotation interval, even just by 50%, to go to 8 to 10 years without significantly increasing our losses to Fusarium wilt? And one of the questions I got from a grower early on when I started this job, this grower had traveled internationally. He's also a field rep for a company. He said, why is it that in Denmark they can grow spinach seed crops on a 5-year rotation and they don't see Fusarium wilt? And that was a really good question. As I dug into aspects of soils in Denmark versus here, one of the things that really jumped out is soils in Denmark are alkaline and highly calcareous. And when I dug into old literature, found some papers from the 70s on Fusarium wilt of tomato in Florida. Some work had been done back then looking at agricultural limestone amendment to suppress tomato Fusarium wilt. So we started a series of trials looking at agricultural limestone amendment in growers' fields in Western Washington, fields that had been out of spinach for as little as 5 to 6 years, which normally would be suicide for most of these varieties that are grown for seed production. And you can see this is a photo I took in 2006, the first year we did this, and you can see that highly susceptible female line on a 5-year rotation with no limestone is just decimated. And behind it, we had some different rates of limestone amendment versus a partially resistant female. But even then, that's very, very poor growth. And you can see the male line, there's just nothing left. It's so susceptible. So we've done some amazing work showing that agricultural limestone amendments can help suppress Fusarium wilt. It's a transitory suppression, not long-term, but it makes a difference. So you can see in 2006, we saw this on the highly susceptible female line represented by the blue line over these different rates of limestone amendment and the marketable seed yield at the end of the season. And we noticed this very interesting phenomenon with this partially resistant female, where we increased the seed yield by about 1,000 pounds per acre, not tons per acre. And then the seed yield plummeted dramatically. And as we were monitoring soil nutrient status and plant nutrient status about every 3 weeks, we saw this tremendous decline in certain essential micronutrients, particularly zinc and manganese. And so we subsequently did trials looking at if you make a foliar application of a zinc and manganese, either chelated or sulfate form, at that rosette stage when the plants are starting to bolt, transition to reproductive growth, you can eliminate this negative impact of tying up those micronutrients as the pH is pushed up by the calcium carbonate. So you can see in 2008 how these higher rates of limestone, we continue to see an improvement in yield because we were able to counter that tying up of the micronutrients by putting on those high rates of limestone that was depriving the plant from what it needed, but also continuing to deprive the pathogen. And we had a PhD student, Emily Gatch, did a long-term 4-year trial, well, not long-term, but 4-year trial looking at, can you optimize the use of agricultural limestone to suppress the pathogen? You don't eradicate the pathogen, it's still there. You don't prevent it from growing, but you suppress it significantly. And so she did a trial where we grew spinach in 2009 in a grower's field after 5 years out of spinach. And the grower came back the following year, planted potato, and then winter wheat. And then we came back and planted the spinach trial in 2012. And she put on limestone at the beginning of the 2009 trial, and then again every year around the grower's rotation, and then again at the beginning of the 2012 spinach trial, which was only 4 years after the previous trial. And the limestone was put on at either 1 ton per acre or 2 tons per acre every year, or not put on at all. And what you'll see is the results here of seed yield, which reflects the amount of disease pressure, with tons of limestone per hectare. So this is equivalent to 1 or 2 tons per acre. At the initial trial in 2009, for 3 different lines that were either highly susceptible, moderate, or partially resistant. And again, for those same 3 female lines in 2012, the 4 years later, where we had put on these applications annually of 1 or 2 tons of limestone per acre. And look at that tremendous boost in yield by putting on agricultural limestone. So again, we've not eradicated the pathogen, but we've created less conducive conditions that the inoculum in that soil is less effective. So it's really a good reflection of how inoculum per se, amount of inoculum per se, is not always the most accurate predictor of disease risk. It's also the conditions around that inoculum, like your pH, your calcium, and we can amend that with things like limestone. So this was a wonderful way to demonstrate we do have the capacity to reduce our rotation interval of spinach by optimizing limestone applications and basically doubling the carrying capacity for spinach seed production in the USA. And one of the things Emily did was to document the degree to which this suppression is related to micronutrient availability. Is she grew the spinach fusarium wilt pathogen in these flasks with different levels of these micronutrients, manganese, zinc, and iron. So this is showing how much fungus grew at these concentrations of these micronutrients. And you can see zinc, for example, if you get deficient in zinc, this pathogen does not like to grow at all. Also with iron and manganese. And as the concentration increased, the growth rate of the fungus increased very significantly. And the amount of spore production was also very dramatically impacted by the amount of micronutrients in the soil. So what you're doing by adding agricultural limestone is you are tying up those micronutrients so they're not available to the fungus. You also have to be careful that you now provide the crop what it needs by doing foliar feed, so the crop gets those micronutrients from above ground application without supplying it to the pathogen in the soil. And now a really important question that a grower came up to me early on when I started this position. I remember it was around 2005 or 6, grower said to me, I've had this wipeout in a field that had been out of spinach for 15 years. Why is this field not safe yet? How can you help us know when a field is safe to go back to? I had 2 or 3 fields I could have chosen. This field I thought was safe because it'd been 15 years out. I could have chosen the field next door, and yet it wasn't safe enough. We had a wipeout. And then a couple of years later, the president of the Seed Growers Association showed up at my office with a 5-gallon bucket of soil, and he plonked it on my desk. And he said, Lindsey, put the soil in pots in the greenhouse, and let's plant a highly susceptible spinach line and see what happens. He says, I want to know whether I can go back to this field. And that was the onset of this long series of trials we've been doing now for 15 years to develop a soil bioassay to help growers accurately quantify the risk of fusarium wilt. And so we have been developing this bioassay. We started developing in 2010 as part of Emily Gatch's PhD project. We published the first results of this, the first 5 years of this, in a paper in Plant Disease in 2015. And we've continued to offer this bioassay every year. And you can see in this photo, some of these pots, everything's dying, and some of the things look really healthy. So what happens is the growers bring us soil in pots. We process that soil. We plant it out and replicate 12 pots in the greenhouse. 4 of them get planted with a highly susceptible female, 4 with a moderately resistant female, and 4 with a partially resistant female. We randomize it in the greenhouse, so we're blind. We don't know what we're rating. And then we have an open house in January when we unrandomize everything. So the grower can come and take a look at the 12 pots of that particular field. And here you can see that early on we used to do 5 replications. Now we cut it down to 4. But here's the pots that were planted with that partially resistant female, the moderate, and the highly susceptible female. Everything died. This is a grower's field that they were considering planting that year. And this is the classic high-risk scenario. We get some intermediate risk fields where like soil B, where this, if you have some good resistance in the male and female line, you could be okay. But if you've got high susceptibility, like we saw in some of those photos I showed you, you're going to have a wipeout. And this shows you one of the aspects of risk, besides looking at the bioassay and what's in the soil and the conditions of that soil for conduciveness to the disease. A huge part of that risk equation is the susceptibility of the lines that growers get asked to grow. And most of the time, these are proprietary inbred lines that are confidential. They don't know what they are. They don't know the genetics. They don't know the level of susceptibility. And that's a huge part of the risk equation we've been trying to quantify for growers. And sometimes you'll see soils like this soil C, which is perfectly safe, no matter how susceptible that female line was, everything grew really well. And this is what we've been trying to quantify for growers. And I'm showing you the results of the first 2 years of the bioassay that we did for the growers. In the top figure from 2009-10. So we start the assay in December, we finish it in January, February. So growers have results by March 1st, which is the date when they pin on the map where these crops will be planted to avoid pollen cross-contamination. And you can see the Fusarium wilt index 1 means everything died in the pot. The green bars represent the pots planted with the partially resistant spinach line, the orange, the moderate, and the red, the highly susceptible line. And you can see that first year, a large percentage of the fields we tested were highly susceptible, very high risk if they were planted with a susceptible line. And every one of these fields was being considered for planting that next year. And based on the success of the trial, you can see the second year we had a tremendous increase in the amount of soil samples that we received. And you can see a lot of those fields are very safe, but you still have a significant number of fields that were very high risk if either the male or the female is susceptible. You might say, well, what if the male dies? Is that not an issue? It is an issue because the male dies, the female, when female plants are not pollinated, they will sex revert. So they'll start producing their own pollen. Now you have a bunch of inbred seed. You can't sell it. It doesn't have the correct genetics. The female dies, then clearly you're not going to have seed to harvest. So this is why both male and female lines need to be able to grow and to have a marketable spinach seed crop. And we did a lot of validation of the bioassay using other tests, going out to these fields, planting these same 3 female lines in rows, seeing if the results in the field reflected what we saw in the bioassay. And we had very strong correlations between field results and the bioassay results for these different soils. And again, just different data that's in this paper published in 2015, validating what we're seeing in the greenhouse to make sure what we're observing is reflecting the actual Fusarium wilt risk out in the field. One of the questions we came up with, can we just test all kinds of properties of these soils? We looked at pH, we looked at various micronutrients, macronutrients, we looked at organic matter, cation exchange capacity, EC, we looked at years out of spinach, we looked at texture of the soil, and we looked at quantifying just Fusarium oxysprum, not Fusarium oxysprum spinaceae, because there is no way to test soils directly, and there wasn't at the time. And also Verticillium dahlia, which is another Fusarium fungal wilt pathogen that can infect spinach. So in all these soils that we test, we also run all these other tests. And we ran a regression analysis for the first 121 fields in those first 5 years to see if we could get a strong prediction of the quantitative risk of Fusarium wilt. That's Y, which is your risk of Fusarium wilt. And then these models, you can see the different variables that have the biggest influence on how much disease are things like number of years out of spinach, soil pH, sometimes it's texture, depending on the parent line, sometimes it's clay, sometimes it's ammonium, which, alkaline soils suppress and so on. But you can see the R-squared, which is a prediction of how strong you correlate these variables with the yield loss to Fusarium. You can see it is very poor. It's 0.33, maximum is 1. So you've got at most a 34% accounting for that risk of Fusarium wilt with all these variables that we try to plug into the model. So really, none of these variables alone, or even in combinations, comes even close to quantifying that risk. So we still need to do this fairly tedious, labor and time-intensive bioassay to get an accurate prediction of risk. And a really important feature I want to point out to many of you, even though you may never grow spinach in your life, is I see a lot of people using soil testing labs that offer the ability to test for Fusarium risk. And very often they are presenting just Fusarium to genus, or sometimes to species, Fusarium oxysprum. And I think it's really important to recognize that there are many, many different Fusaria and Fusarium oxysprums in soil, and many of them are not pathogenic to the host plant that you're growing. Many of them are even beneficial. So getting a soil test result that just tells you how many colonies of Fusarium you have per gram of soil, or how many colonies of Fusarium oxysprum, is completely meaningless when it comes to knowing disease risk. You have to know, in the case of Fusarium wilt, you have to know what the subspecies level is that's pathogenic to the plant that you're growing. So we looked at, for example, in 2018, here's all the soils we tested in 2018 from lowest risk to highest risk on that susceptible female. So again, that's the severity wilt index at the end of the season, the bioassay. And our check lines at the end here, check soils at low, medium, and high risk. And this is the same soils with the amount of Fusarium oxysprum that we quantified by just plating that soil out on an agar medium and Petri plates that allows you to quantify Fusarium oxysprum, but doesn't tell you if it's spinaceae. And you can see there's zero relationship between the amount of Fusarium oxysprum in the soil and the amount of fusarium wilt risk. And we have done this almost every year, actually not 14 out of the 15 years we've tested soils for the bioassay. We have shown there is no significant correlation between testing soils for fusarium or Fusarium oxysprum and knowing the risk of spinach fusarium wilt. And I see growers in Washington State who, especially in the Columbia Basin, do grid sampling of entire fields on a 2-acre basis. So every 2 acre is represented by a soil sample that gets tested for fusarium. And I say, for example, for onion growers, and they get a test back that says X amount of fusarium per gram for each 2-acre grid. And they're making decisions around things like fumigation, which is not an inexpensive treatment. And yet those results are meaningless because there are so many fusaria in soil, they're ubiquitous, and there's so much diversity and even beneficial species. So this is a really important consideration to think about when you're making decisions around soil, biological soil data and risk management. I mentioned too that we also test these soils for Verticillium dahliae. It causes Verticillium wilt of spinach. It's a very important pathogen because it can be seed-borne. And this is just one example of soils we tested one year with the amount of Verticillium dahliae, which is a pathogen, and Verticillium tricorpus, which is not a pathogen of spinach. So again, making sure that labs that offer these kinds of testing can differentiate the pathogenic species or subspecies from the non-pathogenic. And you can see some very high levels of Verticillium dahliae. We've even had fields up over 200. And for a crop like potatoes, the threshold is 0 to about 30 or 40 CFUs. So when you see some of the extremely high levels that are coming back here, and you see some of those fields that do get planted to spinach, you can see what happens with the spinach seed crop in a field that had 120 colonies of Verticillium per gram of soil. This should be bright green, thriving crop, and it's showing this classic Verticillium wilt symptoms. It's not fusarium wilt. It won't totally kill your crop. You still get seed, but you get a significant reduction in yield if you have over about 50 CFUs of Verticillium dahliae per gram of soil, and you get infected seed. The harvested seed will be infected. So this is another service we offer with this bioassay. Now, I mentioned to you that part of your risk for a seed grower is not just how much inoculum is there and the amount of disease pressure, but also how susceptible are the parent lines you're growing. So one of the things we now offer for growers and have for about 8 years for seed companies is to test the level of susceptibility of those parent lines that they're contracting to seed growers to plant. And we now know, and we didn't at the time we started this, but we now know from a PhD student in my program that there are 2 races of the spinach fusarium wilt pathogen. So we actually now screen for responses to race 1 and race 2. So these are data from one of the years of testing all these spinach lines. This is disease severity index against race 1, either in a high risk where we have a high level of inoculum in the soil represented by the white bars, or a moderate level of inoculum in the soil represented by the black bar, or no inoculum represented by this tiny little gray bar. And you can see for race 1, these fields on the left side, so for example, inbred 3, sorry, not fields, but parent lines, inbred 3, had really good resistance to, here's growing in the control with no inoculum, with race 1 at medium and high rates of inoculum, with race 2 at medium and high rates of inoculum. So you can see no bar on race 1 because it had no disease when in the presence of race 1, regardless of the rate of inoculation, but quite susceptible to race 2. If you look at inbred 8, it had some partial resistance, inbred 8 to race 1, and maybe a little bit of resistance to race 2, and you can see that's reflected in these photos I took at the medium and high rate of inoculation with race 1, medium and high with race 2. If you look at inbred 17, highly susceptible to both races, everything died even at the moderate rate of inoculation. So this has been important information for seed companies to try and match up the more susceptible lines with the lowest risk fields that we detect in the bioassay. And so we offer this every winter. We now charge growers $250 per field, or companies $200 per parent line. We give them the complete soil nutrient analysis. And in late January, I have an open house where each grower can come and take a look at the parts for their field without other growers around. And I have this incredible opportunity every year we offer this bioassay to have a 30-minute conversation with each of these growers about the risk we see represented in the bioassay, what might have contributed to that risk in terms of crop rotation and other practices, and how they're making decisions around management based on the information we provide them. And that's been an amazing exchange of expertise for me to learn from growers and for growers to learn from me as a plant pathologist. And this has been one of the most valuable aspects to me as a plant pathologist with this bioassay, is to have this direct dialogue and exchange about risk every year that we offer the service to growers. And as I said, we now do this as well with parent line screening. And we have tested over 600, almost 650 fields over the last 15 years. And we've published another paper looking at the lessons we've learned from 10 years of stakeholder adoption of this bioassay. So we're now up to 15 years, but that paper was published a few years ago. We also had a question, as we've been looking at these limestone amendment trials and doing all this work with spinach fusarium wilt, it became apparent to me that some of the difficulty of spinach seed crops in combating fusarium wilt in the soils is soil structure and soil tilth and soil health. Soils in Western Washington are heavier. They get worked pretty hard, particularly for some crops like fresh market potatoes, where you want nice, good soil structure around your potatoes to get nice shaped tubers for fresh market options. And that means the soil structure gets damaged every year from the degree of cultivation of these soils. And I've seen many spinach seed crops where the ground has a hard pan layer, not too deep down, and there's a lot of J rooting in some of these fields. So I wanted to look at the degree to which practices that might help improve soil structure, soil tilth, could also provide a benefit to managing fusarium wilt. So we've done a series of trials looking at compost amendment. We happen to use a biosolids compost because these are non-food, non-feed crops. They're seed crops, so you can use a biosolids compost. The town of Lacona donated a biosolids compost for us to do this work. And you can see us putting the compost on it, treated and non-treated plots prior to planting a spinach seed crop. And so if you take a look here, this is dry weight of spinach per plant in the middle of the season and seed yield at the end of the season in pounds per acre for those same three female lines, susceptible, moderate, and partially resistant to fusarium wilt. The white bars are the absence of any treatment. The green bars are where we were looking at a fungicide called proline to see if it would help. And the brown bars represent the plots that were treated with compost. So in the susceptible female, we saw a 20% increase in seed yield when we applied a single application of compost just before planting. The moderate had a 48% increase in yield, and the partially resistant had a 30% increase in yield compared to the non-treated. So this is a really significant increase, showing how not only looking at limestone amendment, but also looking at quality of the soil and providing good soil structure and nutrition can really help further reduce that impact of fusarium wilt on spinach seed production. Now, one of the key things that we also wanted to look at is why is it that certain strains of Fusarium axisprum are pathogenic on spinach, these former Specialis spinaceae strains, and others are not? What's the genetic basis behind this? So we had a PhD student, Alex, working on this, and we worked closely with folks at the University of Amsterdam that are specialists in Fusarium the genetic basis of Fusarium isolates that are pathogenic on different hosts. And he was trying to figure out, don't have to worry about the details of these two questions, but basically, what is the genetic basis behind spinaceae as a pathogen of spinach? And so he did a lot of work, and I'm going to give you a snapshot of what he ended up showing. He took isolates of Fusarium that we knew were pathogenic on spinach based on running tests ourselves. Also isolates that came off spinach but were not pathogenic. So just because you get Fusarium off a wilting plant doesn't mean that isolate caused the wilt. So he has these non-pathogenic spinach isolates and these spinaceae isolates, we call them FOS for short. And he compared them, the whole genomes of these isolates, with isolates of other, the isolates are pathogenic on other hosts like radish, peas, banana, melons, tomato, cucumber, onion, and so on, banana, and looking at this genetic relationship of these isolates. And what became clear very quickly is we have two very distinct groups of spinaceae. We now know after further testing, it turns out these are races, race 1 and 2, and they differentiate very clearly from these non-pathogenic isolates that we can get off spinach seed, off spinach plants, or soil where spinach has been grown. And Alex was able to go in there, and this is really complicated, but these are 120 different isolates of Fusarium axisprum. Alex's spinaceae isolates are shown in green, and his non-pathogenic spinach isolates in brown. And down all these rows that you cannot read is different, what we call putative effect as potentially genes that might be involved in that interaction between the host and the pathogen. And you can see basically blue means that gene is there, brown, gray is absence, and you have a very unique genetic fingerprint for the spinaceae isolates from all these other isolates and from the non-pathogenic isolates. And when you look up closely at some of these genetic factors that might be driving the ability for spinaceae to infect spinach, here's the spinaceae isolates, race 2 and race 1, and the non-pathogenic isolates. And when Alex inoculated these isolates onto 2 different parent lines, this is our highly susceptible line that we've been using in all these limestone trials and bioassay and so on, and this partially resistant line. What you see is with race 1, the susceptible dies really quickly. The partially resistant line does develop symptoms, but compared to the control, but doesn't get completely killed. But he noticed that these other isolates, which we now call race 2, that actually parent B, this what we were calling partially resistant, is actually more susceptible than parent A. And this is what classifies what we call a differential host or differential varieties that can separate these races. And this has been an important basis of identifying these 2 races of the spinach pathogen. So Alex then went ahead and tested 68 different commercial spinach varieties to see if we see some differences. So this lower figure here on the top, group 1, this is race 1, these 68 varieties ranging from the most resistant to the most susceptible. And these, in the same order, this is their response to the race 2 isolates. And you can see there's no relationship correlation-wise between the response of these 68 cultivars to race 1 versus race 2. This has been really important information to provide to seed companies, to provide to spinach breeders to recognize when they're trying to screen for resistance to Fusarium, they need to be screening for these 2 races separately because cultivars can respond very differently to these races. And this is just an illustration here of, for example, cultivar F. Here is growing in the control, and with race 1 and race 2, everything's resistant. We've got something here, like cultivar E has good resistance to race 1, but not race 2. And cultivar A has no resistance to race 1, but good resistance to race 2. And you can see this consistency across the paper that, and this was published in 2022. One of the really interesting things that Alex noted as he was looking at this and repeating these trials to look at, can he get consistent results when he repeats this? So this is results for Fusarium wilt severity for some of these varieties in the fall of 2020 and also in the spring of 2020. He repeated the trial exactly as he had done it the first time. Now, if everything was consistent, the response would be along those straight lines. So things like baboon that are really susceptible to race 1 in, very resistant to race 1 in spring were also resistant to race 2. Hammerhead, Kiowa, highly susceptible. Doesn't matter which time of year Alex ran these experiments. So you noticed all these black cultivars with black circles are represented along those lines, which means they consistently display the same reaction, whether it was susceptible or resistant or intermediate when the trial was repeated. But there was this really interesting phenomenon with some of these varieties when they were tested with race 2. For example, Hammerhead in the spring looked really, really resistant because it's down on this end of the x-axis. But in the fall, it had a 75% severity, so it looked really susceptible. And at first we thought, okay, something went wrong. Do we mix up isolates? Do we make some mistakes? What's going on? Why are these race 2 isolates, the cultivars, showing these change in response to race 2 isolates? And not all of them. Some of them, like Veriflay, were consistently highly susceptible. This sugar beet line was consistently very resistant. What is going on that these lines are changing their action when we repeated this experiment? And one of the things Alex noted as he looked at all the things that might have influenced this is there were some very distinct differences in sunlight intensity, daylight intensity, when he ran these 2 experiments. So he ran a test to see if the amount of light influences the response of these spinach varieties to the 2 races. And sure enough, he saw a very, very big difference when he grew, say, for example, baboon with race 1 without shade or with shade, very similar responses. Race 2 without shading and with shading. And you can see how every time he shaded, the varieties were more susceptible, particularly to race 2. And again, this has been really useful information to say to growers and to seed companies, the response of a variety to one race can differ depending on the amount of light intensity under which that variety is growing. So for breeding purposes, this has really practical application to trying to screen and identify good resistance, because if you think something's resistant, But then you plant in an area where there's a lot of cloud cover and a lot of rain, it may suddenly look highly susceptible. So another side benefit of this work that Alex has done is he's been able to identify some unique genetic areas of these race 1 and 2 isolates that separate them from the non-pathogenic isolates and separated from each other. And he's been able to design a molecular DNA-based assay that can identify the Spinaceae isolates, because as I mentioned before, we've not had the ability to tell which isolates were Spinaceae or not without actually growing it in a petri dish, growing spinach plants, inoculating, and seeing if they develop wilt. But we now have the ability to test infected plants, to test live cultures of the fungus, to tell is it Spinaceae and to tell which race it is. And we can detect it in the plants as early as 7 days after planting in infected soil. So we now have a tool to be able to tell more efficiently than having to run a 6-week pathogenicity test. And just to wrap up some of the other work we've been doing, we had a postdoctorate, Sanjaya Gowali, who started screening the USDA spinach germplasm collection, as well as wild spinach collections from the Dutch germplasm center, to see if we can find sources of resistance to Fusarium wilt. And we have found some excellent sources of resistance. And this is just complicated genetic data here. You can ignore that. But we found a number of these, what we call plant introduction lines that are in these germplasm collections that had very good resistance. And most of them were Asian spinach types, not your typical baby leaf type of spinach. And one of the very interesting things is Alex Sanjaya looked at the genetic diversity of these lines. The center of origin of spinach is this area here in Uzbekistan, Tajikistan, Pakistan, Afghanistan area, Kyrgyzstan. And as he went back and looked at the GPS coordinates of where these lines were collected by curators for these germplasm centers, you can see that the different colors here represent the genetic groups to which these lines belonged. And you have this very distinct geographic separation of the PIs that mostly were green versus mostly blue. These are Spinaceae turkestanica, wild ancestors to spinach. And this really reflects, there's a big mountain range that runs down, right down here through Kyrgyzstan, Tajikistan, Uzbekistan, that physically separates these PI lines to the north from the PI lines to the south. And over the eons and thousands of years, we believe this has driven some of this genetic differentiation. The really good thing is that Sanjaya was able to find sources of resistance in both of these populations of Spinaceae turkestanica that can now be integrated into the cultivated spinach, Spinaceae oleracea. So we hope that that presents a lot of potential for getting better resistance into spinach cultivars. So to just summarize some of what we've done and working on spinach Fusarium wilt now for almost 20 years is we have demonstrated there is this capacity to double the carrying capacity for spinach seed production in the USA through careful use of some of these management practices. We've shown that highly acidic fertilizers like ammonium can really help, unfortunately, make soils more conducive because of putting acidifying fertilizer right in the furrow at planting. Whereas nitrate, we can't use nitrates in high rainfall areas because they leach. So looking at alternatives such as urea can be really useful for trying to minimize that disease pressure in furrow at planting. I'm just plugging my charger in because my computer's low on charge. Well, thank you. We've also looked at a number of fungicide treatments to see if they can help, particularly seed treatments to reduce the risk of having Fusarium on seed that might contribute. But really the bioassay and the soil amendments like limestone are the most important management practices that we've been able to provide to growers, and screening of parent lines to match that risk to reduce this likelihood of wipeouts. I wanted to mention that a big part of what I do in my job is applied research and also extension. And I try to take that information that those practices we've learned through my job to a class I teach called Field Plant Pathology. It's a graduate course. And a big objective of this course is to get students out of the classroom and into the world of agriculture and plant production, and to learn about managing plant diseases and detecting plant diseases and working with stakeholders in the real world environment. So we take them around the state for 2 weeks in the summer, 1 week in June and 1 week in July, interact directly with people involved in production, regulation, chemical side, the organic side, the phytosanitary certification side from the State Department of Agriculture, USDA, and just really getting the context of all that they've been learning in their classes and how it fits in the real world. And this just helps students really get passionate about their careers as plant pathologists and other fields of agriculture, as they see just how much science can help drive and improvements to agricultural production. So with that, I just want to acknowledge the various agencies that have funded the work I've been presenting, also the seed growers and seed companies, the students, the technical staff in my program who are critical to being able to do this kind of work and make a difference. But I really believe in the importance of interacting with growers in what we do to make our work more effective. So I don't know, Danny, if that's time for questions now.
Yeah, thanks so much. We have a few questions in the chat and some that folks have already submitted while registering. So let's start with a question from Chloe, who asked about those photos you showed with greenhouses full of the soil samples and the bioassays. She asked, why aren't they contaminating each other? And could a small farm replicate this work without the risk of contamination?
Yeah, so Fusarium is not a foliar pathogen, it's a soil-borne pathogen. When we handle these soils, so as I said, the grower brings us soils in a 5-gallon bucket. We put a lid on, we keep the soil sealed. When we're processing the soil, we have a big concrete wash pad area. Every soil gets handled very, very carefully. So we dry the soil out if it's too wet when it comes in to get it a little bit drier. We put it on tops that can be sterilized and washed between soils. Every soil gets passed through a soil shredder to make sure we don't have big clods and clumps, and get it mixed really, really thoroughly. And then if the grower is planning on putting a limestone that spring, we will amend that soil with the equivalent rate the grower plans to use so that our risk reflects what risk they are likely to encounter if they go ahead and plant that field. So we do that in a cement mixer, and after every soil has gone through every piece of machinery, everything gets pressure washed with hot water and everything gets sterilized with alcohol. And so we are extremely careful. I am very particular about who I allow to do this work because I do not want to be contaminating between soils. But the fact that it's soil-borne and not airborne, and we're very, very careful about how we handle this. When we plant the pots, everyone's sterilizing their hands. They're wearing gloves so they can't get soil under their nails. So we're not causing cross-contamination between fields. So we're very, very careful about this. We have control soils that we also randomize and replicate. So if we are causing cross-contamination, we will see it. But it's shocking when you can see, you have one pot with everything completely healthy right next to a pot with everything dead, and they're in that greenhouse for 40 days. We don't cause, I'm very, very careful about watering. We don't let just anyone water the trials. So we have really tight control. But to answer Chloe's question about could a small farm do this in a shed without risk of spreading, I do believe a small farm could do this. It might not be quite as well controlled as we're doing because we're researchers and we're aware of some of these risks. But I think just the fact that the grower showed up in my office and plonked this 5-gallon bucket on my desk, I think you could do this. Take fields you're thinking about planting, put them in a pots or flats. Soil never behaves well in pots, so you have to also be really careful about how you manage and work with soil in containers, making sure you have the right lighting, the right conditions. What disease are you trying to screen for? What conditions make that disease severe enough that you can see this? So we know Fusarium is favored by heat. So 14 days after planting, we up the temperature in the greenhouse, and we can start to see wilt develop usually within 24 to 48 hours once we increase the temperature. Prior to that, we also treat the seed with apron to prevent pythium in the soil from confounding the effects of Fusarium. So we're trying to separate other things that might be going on that soil. We know sometimes we get crane fly larvae in these soils, so we actually drench with an insecticide to avoid crane fly larvae coming out and moving between the pots and killing all the spinach. So we've learned over the years of optimizing this to separate these other things that could confound our ability to quantify Fusarium risk. So it's not as simple as I'm making it out to be. It's pretty complicated, but I think it can certainly be done if you know what disease you're trying to screen for, you know what conditions favor that disease, and how you can create those favorable conditions in your greenhouse or your shed.
Thanks, Lindsey. And someone named Jeffrey asked about the soil pH before and after the lime addition.
Yeah. When we started this work, Jeffrey, we in all our trials, we had plots that we didn't put any limestone on. We had plots that we had limestone on at these different rates. They're randomized, replicated, and we were measuring pH shift before and after. And we basically see if you put on one, our soils typically are on the pH in the fives, high fives, mid to high fives if there's no limestone use at all. And 1 ton per acre will usually shift at about a half pH unit. 2 tons per acre will usually shift our soils in Western Washington. Again, it's highly variable, so this is generalizing grossly. 2 tons per acre will usually shift soil pH for Western Washington soils by about 1 unit. So I usually recommend to our growers that their pH is in the high fives, they should be putting on about 2 tons per acre to get as close to neutral as possible. If their pH is already in the mid, low to mid sixes, I would say maybe 1 ton would suffice. And it's a really interesting question, Jeff. As I look at 15 years of records, when we started, there were almost no soils at the pH in the sixes. Now we have very few soils that are in the fives because of how much growers are aware now of this benefit of limestone application. So it's been remarkable to see this paradigm shift, but it's happened slowly because you have to persuade growers about this value, and it's an investment you're making in the soil. And so it's been remarkable to see this shift towards majority of our soils now coming in already in the sixes.
That's actually a great transition to a question someone else had, where there's sometimes this tension between plant breeders and soil scientists where, soil scientists think that management is the most important thing, and breeders think that having disease-resistant varieties is the most important thing. And I think your research maybe shows a little bit of both, but is really focused on the soil management piece. So maybe you could speak to your perspective on what are the most important factors. Is it variety or is it soils?
Well, in the case of spinach seed growers, they don't get a choice. They get a contract. They never own the crop. It's proprietary genetics. They have no choice in what variety they're given. Now, the field reps for those companies have a choice in where they put the contract for specific lines. And that's why we do that parent line screening, because the field reps for the company can say, all right, Jeff's field over here looks pretty high risk. We were going to give him a contract for this hybrid, but this one parent in that hybrid is turning out to be really susceptible based on your parent line screening. So we want to avoid that field if Jeff's going to have that contract, or we need to find a different variety for Jeff to grow. If you're a commercial grower that you're dealing with spinach, so Alex's work where he screened the spinach varieties, these are commercial spinach varieties, not for seed production, but for commercial production. So if you're a grower of spinach for selling to eat, you now can say, which varieties are resistant, which varieties are susceptible. You can make that choice. And that's where varietal resistance and knowledge of that becomes much more important, because you have a choice. Seed growers don't have a choice. So you can see we both play a really important role, how much you can control. And I always tell growers, what we're trying to do is figure out, what do you have the ability to control? What do you not have the ability to control? And how do you manage risk around that? So a seed grower doesn't have a choice on parent lines. A grower of a commercial crop does have a choice if there's resistance out there. Some diseases are much, much harder to develop resistance, especially when you've got numerous races and struggles around that. Fortunately for spinach, we have 2 races, and 90% of the fields we test are predominantly race 1. And so we really focus on the resistance to race 1 in selection for these fields.
Thanks. So let's switch tracks a little bit and talk about this kind of making data actionable piece. I was really struck between the figures you showed, which demonstrated no relationship between the tests that were commercially available and the work that you're doing in your lab. And I'm wondering, I'm wondering a lot of things, but maybe we'll start with, have you worked with the labs that are offering those tests? And is there any move towards not trying to spread that misinformation or try to unify methods?
I have presented these kind of results quite frequently. For example, the Pacific Northwest Vegetable Association has a big meeting of usually about 800 people sign up for that meeting in the Tri-Cities every November. And I've been asked to speak about soil-borne disease management, and I often will take these data from our spinach work to say, this is a really important aspect to think about, because we have farmers in Central Washington who are doing extensive grid sampling. They're trying to make the best management decision. They're trying to only treat where they see disease pressure with the grid sampling. But when you have these test results that are really meaningless, and I've asked growers before, some of these labs before, you know that these numbers don't mean anything. And well, growers want these numbers and they want to make decisions around them. So we just offer what they want. And to me, that's a huge conflict of interest. And to me, it's abusive. I can't tell them what to do, but they're relying on the fact that growers don't necessarily understand which data are meaningful and which are not in order to sell a service. And so I have a real problem with this. I presented this at meetings where those folks are in the audience, and I just keep hoping that maybe at some point their conscience will get the better of them.
Yeah, I guess that opens up sort of a larger discussion about both soil testing and also soil inoculants, which I know a lot of people are really excited about, and there's a lot available on the market. So maybe you could say something about your experience and perspective on soil inoculants, soil microbial inoculants.
Yeah, I don't have a lot of work with experience on things like legume rhizobial inoculants, but I've done a lot of work with microbial inoculants for disease suppression and a lot of work on mycorrhizal inoculants. And we all, hoping that there's a myriad of products out there and there's just an increasing number of products because it's very easy to develop these products. It's very easy to get them registered. It's very easy to claim effects on diseases with no data to back it up. And unfortunately, I've spent over $500,000 on research trials trying to evaluate products in an independent way where there isn't this conflict of interest with the product company that's selling them to see if we can find things that really work. And in the vegetable cropping systems that I've tested these products, and I have not yet been able to find good efficacy. And it's a real shame, because not because I'm against this, but I don't want growers to be buying and using products that aren't giving them what they think they're getting. The brochures, the marketing is really slick. They throw all kinds of science in there. It's easy to show in a petri dish that something works. I can make table salt look like an amazing fungicide. It's a real shame, and it's a real frustration, and it's, to me, another one of these conflicts of interest that is a significant problem. Yeah.
Okay, we have a number of questions, but only a few minutes left. So I think I'm going to just end on this question. Your research is so kind of integrated into the grower community there, where growers are really relying on the work that you do to make decisions. And I'm wondering if you could give any advice for either new extension professionals or farm advisors about how they can increase their grower outreach and grower engagement.
Yeah. Danny, when I was hired in 2000 in this job, I had never seen these crops in my life before. And I was terrified of the fact that I was supposed to be providing recommendations and diagnosing diseases on crops I'd never seen in my life before. And it terrified me. And one of the first things I did was, once I got to know some of the growers, and I literally invited growers to breakfast with me. I paid for the breakfast. I did this for years. Every fall, I would bring a group of growers, seed growers, together in Western Washington. I'd do it also in Central Washington. Say, please meet with me. I'm going to talk about what I've seen. I want you to tell me what you've seen. I want to discuss research needs. I want to learn from you, so that when I'm thinking about writing a grant proposal, thinking about a research project, I can base it on your real world knowledge of the crops, how they're grown, the constraints around these practices I'm considering evaluating, what might be real, what might be translatable, what might be ivory towered. And I did that for years, and I paid for that out of my own pocket because I thought I have to base my ability to do research and to make recommendations on their understanding of the crops. And when growers and field reps for these companies realized I respected their expertise as growers or as field reps, and I brought to the table my expertise as a pathologist, that together we're going to make good management programs that are going to be usable, going to be economically viable, but independently, I'm never going to be able to do that. It was the best thing I ever did. I did it out of fear. I did it out of realizing how little I knew and how dangerous that was. And I'd say that's one of the most important things you can do, is acknowledge when you don't know things, seek out expertise, bring those growers to the table and make them feel that they're valued, because they now have a vested interest in seeing your work succeed, and they will guide you, then they'll go to bat for you. And that, to me, was one of the best things I ever did, even if it's out of sheer desperation.
Well, thank you so much for those words of wisdom and for your presentation today. For those of you in the audience, this will be recorded and put up on YouTube, so you can access it probably as soon as early next week. Molly will respond to everybody who's registered with both the recording and all of the resources that Lindsey has provided in terms of her publications and other resources that may be useful. So again, thanks so much, and we hope to see you guys next week, Wednesday at noon, for talking about soil physical indicators, specifically soil moisture monitor sensors. Thanks so much, Lindsey.
Thanks, Danny. Wonderful series you're putting on.
Awesome. Thank you.